Well, firstly, apologies, because it's been a really busy couple of weeks, so rather than putting together a brand new talk and pitching it at a real sort of psychology audience, it's the one that I gave in New Zealand, and initially it was given to some psychologists, but also people in English and history and law, and kind of more book-based people who are maybe a bit graph-phobic. So it's got lots of nice pictures that are nice for this time of day, but maybe slightly lighter on data than I normally would go with. So yes, I've been in Bangor for the last 5 years now. I've done my master's and my PhD before. It's really nice to come back to JMU again. And as Francis was saying, I was working with Steve Tipper, and Steve came here not long ago to give a talk and really stressed that what he's always been interested in is this automatic conversion of vision to action. So initially sort of low-level visual attention processes, but more recently in the last 10 years, this in terms of social interactions and the way that other people's actions, for example, or other people's social cues we receive as visual input are automatically transformed into action in the observer. And at the centre of this is really this idea that we live in this sensory motor world, that right from day one, all our experiences of the world are sensory and motor, of course, in nature, so they're all about incoming sensory input, about the performance of the motor actions. So, you know, how do I learn that this thing over here is a cup? Well, I learn about it by performing actions on it. I learn about its properties through tasting it and smelling it, and the kind of rewarding feelings that I get when I receive what's inside it. So this kind of not just five senses, not just actions, but also proprioception and interoception as well. So we have all these physical experiences that tell us about things like objects in the world, about interactions with other people. It tells us about concepts and ideas and situations, and also about emotions as well. And so these experiences are all very sensory and motor in nature, the actual experiences themselves, and the argument is that their representation is as well. So this is Stephen, one of our two lovely interns in New Zealand, having a cup of coffee. And Stephen is, of course, having a sensory experience. So this incoming information is being represented in these sensory motor areas of his brain. Now, the idea at the centre of an embodied or simulation theory of cognition is that actually when Stephen thinks about this cup of coffee, the same areas of the brain are actually representing that prior experience. So this is really at the core of this idea of embodied cognition, is that all sorts of cognitive processes involve reactivations of the state that we were in when we first had that same experience. And that goes not just for our understanding of kind of objects or concrete things like cups of coffee, but also for things like more abstract concepts, and also for understanding other people. So how is it, for example, trying to remember the order of my slides, you know, how do I understand how somebody feels? Well, of course, I've felt like that. I've had that sensory experience. I know what the internal feeling of being unhappy is like. Is it that this visual incoming image automatically activates in me a simulation of that person's current physical state, and through that simulation I then have access to their internal state? Is this how we can really, and I mean this is one of the most amazing things about the human brain, is the ability not only to respond very quickly to what somebody is doing, but also to be predictive of it as well. Of course these guys will have practiced doing this in the past, but you know the idea, this is what is underpinning the fact that we're so good at fluent joint action. But also just the general complications of social interaction. We can't have social interactions without being able to make predictions of what people are about to do, not just responding to what they do. And this, of course, means, you know, you're assuming these people have some knowledge of one another, that we build on our experiences of people, and we use this to make predictions about how they're likely to act. And we were asking questions about the extent to which this is actually underpinned by reactivations of prior simulations of those people. So when we simulate an individual and we take on a partial representation of their current state, is this being imprinted into long-term memory? Does it exist alongside our memory for the identity of that individual? And is a retrieval of that simulation actually part of our ability to act predictively? So what I should say is, please stop me at any point if I'm not being clear about something. I'm moving through some bits a little bit faster for this audience than I would do with a more general one, but if you're not sure if I'm being unclear about anything, just stick a hand up or just throw something at me. So what's been really novel about the way that Steve has thought about social simulation, and I'll come on to some examples of this idea of embodiment or simulation of other people, but Steve's work has been a lot to do with this long-term representation. So going beyond the kind of online simulations, rather than just thinking, you know, why would it be that, for example, when I see Francis smile, this might cause activity in that rare event, This is my cause activity in my own facial muscles. What's that doing? What's it providing with me online right now? But also thinking, how does this work in terms of actually changing my long-term representations of Francis? How much simulations underpin my ability to better predict in the future what Francis is going to do, or just to change my attitude towards Francis, which of course is a really important thing, not only for fluent interactions, but also just for survival. I don't pick Francis as a particular example for the case of survival, but this is the idea, is how the simulations not only influence interaction, but also influence long-term representations as well. So probably the most famous study, I mean, this was just a slide for folks at Massey, the monkey mirror neuron findings in the 1990s. You know, is there actually a neural underpinning for this idea that cognition may be embodied beyond, the cognition may be represented in areas of the brain that are actually dealing with sensory information and with motor information? Of course, this is the famously accidental finding, mapping up the premotor cortex of a macaque monkey, having the monkey perform trained actions on objects. When one of the experimenters interacts with the object while the machine is still recording single neuronal activity from the premotor cortex, populations of neurons in the premotor cortex are firing both when the monkey is performing an action and when the monkey watches a person or another monkey performing the same action. As long as, in the case of monkeys, there is an object in the scene for the person to act upon, so it's not a pantomime action, it's an actual goal-directed action. And what's found is that there are populations of neurons in these areas that previously were thought just perform motor actions, not have anything to do with representation, that are representing the actions of other people, of other monkeys, of other humans. They fire when a monkey watches a performance on a screen, so they watch somebody else reaching out to pick something up. Some of these are very action specific, some of them are angle specific, so they depend on the viewpoint the monkey is watching the action from. But the key idea being that this is evidence of the kind of first evidence at a neuronal level of a representation of other people's actions in an area of cortex that was thought not to be representational, was simply thought to be about the performance of actions. This has then been followed up with a whole plethora of research, largely using fMRI, as a means of trying to find a similar system of neurons in human beings. Of course, we can't do single neuron work, or largely can't do single neuron work. So this is actually, I'm using some of Steve's past research here. This is something that was done with Paul Downing, one of his PhD students. It's a classic kind of study where I was actually a participant in the study, and it was really uncomfortable. You had to lie in the scanner, and then you had this tray put across your stomach, and your head is kind of clamped in place. And there's an object made out of FIMO, another wonderful contrast of like £1 million worth of scanner, and this sort of £3.50's worth of tray and FIMO object. And you were trained to perform either a lifting action, pick the object up, or a slapping action. This is for a kind of further extension of this study I want to talk about today, using multi-voxel pattern analysis. But the basic point is that you either perform the action or you watch a video, either from the first person or the third person perspective, or somebody else performing those two same actions. So you have, just like the monkey, this viewing and this doing, and you can make a contrast of these two conditions. And if you look at the analysis, this is just a flash of the brain really, but these are areas of premotor cortex and somatosensory premotor cortex, somatosensory cortex on an inflated brain. Basically, the areas in red are areas that were found to be representing not just the participant performing the action themselves, but also to be representative of viewing somebody else performing the action. So they're active in both conditions. And it's just one of the large number of studies that have been investigating this idea of a mirror system that exists both in, not just in cat monkeys, but also in humans as well. This idea that we are representing other people's states with our own sensory and motor cortex. This is another piece of research done by Wendy Adams, who is a PhD student with Steve, also Patrick Bach, I think Indian Morrison on the scanning study as well. This is looking at representations of other people's somatosensory input, particularly of other people's sort of painful actions. So participants in a range of behavioural studies and also an fMRI study watch hands reaching out and interacting with objects, and the hands either grasp the object or miss, and the objects are either this category, which are noxious objects like broken glasses, stinging nettles, knives, or they're non-noxious and neutral objects, things like oranges, glasses that aren't broken, a piece of stone or ketchup packet. So they're either safe things or they're things that are going to induce pain. And the comparisons are basically, there are various attentional manipulations amongst the different tasks, but the important comparison is what is the brain doing when it watches somebody reach out and grasp hold of something that's going to cause pain versus something that's not going to cause pain, or an interaction where the hand is not actually grasping the object. And the interesting thing is that if you provide stimulation to the finger of one hand of the participant using a little vibrating hearing aid while they're viewing these trials, so they're watching these reach and grasp or reach and misses, you can, by carefully tailoring the amount of stimulation to a particular threshold of the participant, you can measure their sensitivity to detecting stimulation, which is given on random trials. The participant responds when they feel stimulation. And this, for those trials where the participant is responding to, is seeing rather, a hand reach out and grasping a noxious object, the sensitivity to the bias towards responding to stimulation is increased, so the participant is effectively more likely to report that they felt stimulation than they are in trials where they're viewing a hand either missing an object or interacting with a non-painful object. And it's evidence that this is somatosensory representation in the participant of somebody else's, in this case, painful interaction with an object. So you're viewing somebody who's about to receive painful stimulation to their hand. This appears to be represented by the participant. And similarly, in functional analysis of these kinds of tasks, this is basically the participants doing the same kind of tasks in the scanner, and when they're watching the painful grasp condition, we can see this representation in somatosensory cortex. So this participant actually, or these participants actually appear to be representing the painful grasps of another individual who they're watching, reaching out, interacting with noxious objects in the area of their cortex that would represent that kind of sensory stimulation were they to be feeling themselves. So it's just trying to, like, this idea that the understanding of somebody else's internal state, the ability to understand how somebody's feeling, how they're going to affect the environment, potentially what their actions are doing, or what their next action may be underpinned in this sensory motor representations within the viewer. And this is where we're kind of centered in terms of the work in my thesis. And Steve and Patrick Back did a lot of work on motor compatibility effects. So in these kinds of tasks, participants are being asked to make hand or foot responses, and they have to make these responses whilst viewing videos of, for example, somebody kicking a football or typing on a keyboard, and they may, for example, have to identify who the person is or what the action is that they're performing. The important thing with these kind of sets of studies is that if you're asking me to make a foot response whilst I watch somebody kick a football, I'm going to be faster making that response than I am if I'm watching somebody typing on a keyboard. And if I'm watching somebody typing on a keyboard, I'll be faster making a hand response than if I'm watching somebody kicking a football. So actually perceiving the action is speeding my own reaction, and seems to be suggesting that the other person's action is being represented in motor regions and then facilitating the performance of an action with the same limb. But what was interesting in terms of the way that Steve and Patrick looked at this was to look at the actual way that these representations might be involved in our kind of whole representation or long-term representation of an individual. So if you ask participants to make similar categorization responses with a foot or with a hand, whilst they are responding to people who are known to be, for example, tennis players or footballers, you get this same facilitation effect or same compatibility effect, where hand actions are going to be faster if made to a tennis player than a footballer. Foot actions will be faster if made to a footballer than to a tennis player, despite the fact the images are presented so you don't actually see the person performing an action, you don't even see the limbs that they would perform the action with. And what it suggests is that our representation of these individuals actually has with it kind of tied up this simulation of the actions that we're used to seeing them perform. So my idea of Wayne Rooney as a footballer seems to involve some kind of simulation of the process of playing football, at least the process of repetitively performing foot actions. Similarly, Steve and Andrew Bayliss, who's now down in UEA, did a lot of work on gaze cueing. So gaze is a very salient social cue. It tells us where somebody's attention is allocated in the world, which provides us really important information about what's important in the world, about where that person's attending. It guides social interactions. Gaze is something that we kind of follow without an ability to really consciously control what we're doing. So if we see somebody gazing off to one side of the room, our gaze automatically follows their gaze to that side of the room. It's not something that we really have any control over. It's a very powerful salient cue. In a classic gaze cueing paradigm, a face will appear, it will gaze off to the left or off to the right, and the participant's task, they're told, don't worry about the faces, you can just ignore them. You've got to categorize an object that's going to appear either to the left or the right of the face. In the case of these tasks, the object's either a kitchen object or a garage object, and your task is just to press one key if it's kitchen and one key if it's garage, as fast as you can do. In trials like the top trial, which are congruent, the face gazes in the direction where the target is about to appear. In incongruent trials, like the bottom one, the face gazes in the opposite direction. In these trials, participants will respond faster because their attention is already over on this side of space, basically where the target's about to appear. Now, in the experiments that Patrick and Steve did, that Andrew and Steve did, the important thing is these faces have contingencies. So some faces will appear, and every time they appear, they gaze in the opposite direction to where the target appears. Other faces always gaze in the direction that the target is about to appear. So some faces always cue the participant, some faces are always unhelpful and they cue them in the wrong direction. The participants are actually unaware of the fact that they report a triangulated interview at the end, they're actually unaware of these contingencies. So although they're very conscious of the fact that the eyes are gazing, they're not actually conscious that certain faces are helpful and certain faces are unhelpful. But at the end of the task, the participants are then probed and are asked to make choices between two faces, one of which will always be one of these predictive valid faces, so one of these people here. One of them will always be a predictive invalid face, one of the faces that looks in the wrong direction. And they're asked things like which one of these faces is more trustworthy, which face would you prefer to see. And this is a plot of the choices of predictive valid faces. So basically, the predictive valid faces are significantly more likely to be chosen for being more trustworthy or for the participant preferring to look at them. So what's actually happening is, although the participants are unaware of the contingencies that are assigned to the faces, their actual attitude towards the faces, their long-term representation, is being changed by a sharing of a body state with that particular individual and the outcome of sharing that body state. So Kathy looks in that direction and I look in that direction, and that slows me down. Something is marking down in my memory, whether it's the affective response to that or whether it's something to do with just a memory for what happens when I simulate Kathy's body state. But it's changing a long-term representation of Kathy as an individual. So in the current set of studies, in the first set of studies in my PhD, we were really interested in, does this kind of long-term change happen in terms of emotions? We respond to people's emotions online, so if I see somebody smile, I understand that they're feeling happy, but obviously I also need to be able to make predictions about how people are going to act. Some people are kind of safe to approach, some people are less safe to approach, and I need to be predictive of their oncoming emotion rather than just responsive. So I keep saying I need to retire Alex Ferguson as my example because he's retired, but Alex Ferguson, the football manager, is famously a really grumpy person. So when I experience Alex Ferguson, I see him, he's really grumpy, and I see him, he's like having a go at somebody, and I see him and he's bawling his head off at somebody. Every time I see Alex Ferguson, I'm seeing this angry expression, and I'm understanding that this person is angry. And something is supporting that process of understanding how he's feeling. But the important thing is, how does that actually change my representation of Alex Ferguson? So when I meet him in the future and he's in a neutral state, I need to be able to predict, this is a guy who gets angry, this is somebody who it might not be safe to approach. And this simulation somehow involved in this ability to be predictive rather than just responsive. So to investigate this, we've been using facial electromyography or facial EMG, so we can measure very slight movements of the facial musculature using small electrodes placed over sites of interest. And specifically for us, because we're interested in these happy, positive, and angry, negative emotions, we measure activity from the corrugator supercilii in the brow and the zygomaticus major in the cheek, and we can measure activity that occurs at a level that isn't actually, you wouldn't perceive it, it's not visual. Obviously you can measure proper smiles and proper frowns. We can measure very, very rapid and very, very slight activations of these two muscles. Now, if simulation is involved in emotion comprehension, what we should see is some sort of simulation of somebody else's emotion when we see them in real life or when we see them on a computer screen. And there's an effect that's been well replicated in a lot of the literature, an effect called the facial mimicry effect. If you present an emotional face on screen to a participant, you see congruent activity very rapidly in the corresponding muscles. So seeing somebody else smile will activate the zygomaticus major, seeing somebody frown will activate the corrugator supercilii. And this activation occurs very rapidly, so within a few hundred milliseconds. It appears to be automatic and the participants can't control it, they can't seem to completely inhibit it, nor are they aware that it's happening, nor does it appear in most cases as a sort of visible expression. It's actually just a very rapid and slight activation of the muscle. But it's a sign that when we actually see somebody else's emotional state, we're simulating that emotional state with our own facial musculature. And this was the effect that really in the first half of my PhD we were interested in how we replicated, and in fact this is data from our replication. So when we present a fixation cross followed by the presentation of the emotional face, if that emotional face is happy, we see more activation in the zygomaticus than if the emotional face is angry, this blue dashed line. And in the corrugator, we see the opposite. If the emotional face is angry, we get more activity in the corrugator than if it's happy. And this division is occurring within a few hundred milliseconds of the onset of the stimulus. So we're seeing this very rapid facial mimicry, this very rapid simulation of somebody else's emotional state. Now the question is, firstly, what is that doing? One of the suggestions is that this is what's called a facial feedback system. So if you do, for example, manipulate the position of somebody's facial muscles, so in the classic, I think it's a strap step, I think they were called. If you place a pen between somebody's lips, then you sort of inhibit the activity of the zygomaticus, and you find that your brow draws down, put the pen between somebody's teeth, and this activates the zygomaticus. You can do a similar thing with the corrugator by getting people to draw golf tees together on their foreheads. And in these kinds of studies, what has happened in the past is that people have said to participants, they've presented them with basically neutral stimuli. They've asked them to provide effective ratings of, for example, fairly neutral cartoons. And by subtly manipulating the participants' facial musculature, they can actually change their affective responses to, in this case, for example, cartoons, but other studies have looked at this in terms of the ability to determine somebody else's facial expression, for example. And actually changing the physical state of the face of the participant changes their affective state, their mood state. So there seems to be this bi-directional relationship between the affective state and the physical state. So by manipulating the physical state, you can manipulate the affective state. And we all know that when we feel happy, for example, we smile. When you look at studies where participants have been people who have had Botox injections, you see evidence that supports a similar kind of idea. People who have had their brows denervated so they can't activate their corrugator supercilii have been shown, for example, to be worse at determining sad and angry facial expressions, so their emotion comprehension is poorer. They're also not as good at determining negative emotions in prose, for example, in sentences. So there seems to be a role played by the facial musculature in supporting the representation of other people's emotions, of other people's facial expressions. So simulation is suggested to be supporting our ability to understand how somebody feels. So what potentially is going on in our facial mimicry effect is that these activations online during an interaction are actually somehow supporting my ability to understand the person I'm interacting with's internal state and understand their mood state. So the question then is, what happens if we then meet that person again in the future, having simulated their state in the past when they were emotional? What happens if we meet them in the future and they're in a neutral state? Do we then reactivate that simulation again? And is that part of being able to predict how that person is likely to act? So we changed the classic facial mimicry task in the case of our studies. We start off with an encoding phase. So participants in this case, all female participants, the important thing in this stage is that participants see faces that become expressive. So all the faces they see either smile or frown. But the key thing is that a particular identity will either always smile or always frown every time it appears. Participants get multiple exposures to each identity. Half the participants in the data I'm presenting here have the task of identifying the emotions. They just pressed, is it a happy face or is it an angry face? Half the participants had a task of identifying oddball trials, where the face actually changed identity partway through becoming expressive. But the important thing is that all the participants see, after a fixation period, a 1500 millisecond presentation of a neutral face, followed by a rapid morphing of that face, a kind of video that looks like a naturalistic expression, into either the smile or the frown, and then the face remains on screen in its emotional state again for about a second and a half. And as I say, the important thing is the participants are getting this experience of every individual having a consistent emotion, either always happy or always angry. And this is where we predict, firstly, that we should see facial mimicry effects, but also that learning should occur. A few minutes after they finish this task, when the data's been saved, they're then moved on to a second task where
Almost all of the trials they see are a face that they saw in this task, but presented just neutrally as a static neutral version of the face. The participants who did the identity task now do a task where they look out for oddball new faces. The participants who did the emotion task now look out for oddball emotional trials. But the key thing is most trials are neutral static faces, so any activation we see of the facial musculature is actually occurring because of something that happened in the encoding up here, and not because any mimicry is going on in this section of the experiment. So our prediction is here that we should see facial mimicry, and we would predict that this simulation is then encoded for retrieval later on. We would predict at this stage, if you're exposed to somebody who you previously simulated, that we should see some reactivation of that simulation. All the time, the participants having facial EMG recorded, and looking at the data from the facial mimicry section and encoding section. In the zygomaticus, we can see consistently that zygomaticus activity is greater to happy faces, so this blue dashed line, than it is to angry faces. And corrugated superciliar activity, the opposite, is greater to angry faces and happy faces. So we replicate again our passive facial mimicry effect. We see this across all 4 blocks. This is very consistent. In fact, in this data, and I'm not going to show it on this graph, but there's an interesting effect as well, where in the later blocks, the participants start to activate this mimicry earlier on in the trial. So the
averaged over quite a few trials.
Averaged over 64 trials. Sorry about the bouncy screen. So, yeah, it's pretty annoying. The zygomaticus activity, interesting, there's a slight temporal difference between the two. The corrugated mimicry seems to come on very rapidly when the face is first viewed in its emotional state and dies off in this rest screen afterwards, or a couple of seconds here after the face is presented. Whereas the zygomaticus activity appears to have more longevity, so it lasts into that rest period. But also interestingly in the zygomaticus, when we look at data collapsed together from a series of similar experiments, we can see this actual predictive retrieval of the facial mimicry. So in this stage where the face is neutral and it's going to become emotional, in the later blocks, the zygomaticus actually starts to become active in response to seeing these identities that presumably the participants are now predicting this face is going to show this happy expression.
Do you segment these populations on any personality scales of empathy, etc.?
We did look at AQ, and we looked at anxiety, but we didn't get much variance in our sample, presumably because it's all female undergraduates, similar age groups. I don't think we had enough sensitivity really for EMG measures. But I mean, it would have been a prediction of one of our predictions initially shown in the past that we should see some division, but those studies that have done that with facial EMG have had especially selected populations to run. So the key question is then what happens in the retrieval phase? Interestingly, in the corrugator, where we get that transient mimicry in the encoding phase, we actually see no difference. And here we've split our participants, so we're looking at those participants who did mimic versus those participants who didn't mimic. Okay, so in the first, we take our data from the first section of the experiment, we say, let's look at the participants who show the facial mimicry effect, because according to a simulation hypothesis, if you're retrieving simulation, you need to simulate in the first place. So amongst those participants who actually show no simulation, we wouldn't expect retrieval. In the corrugator, as I say, where we saw this transient mimicry, we actually see no retrieval of those simulations, and I'll have to discuss at the end why we think that would be, but there isn't really time for it now. But in the zygomaticus, you can see that while as in the non-mimickers, we don't see any difference between faces who previously smiled and faces who previously frowned. We now get this strong reactivation of the zygomaticus in this second period where the faces are all neutral. So this activation must be driven by an encoding of that simulation that occurred in the first half of the experiment. And this is really the kind of novel aspect of this experiment, that nobody's really looked at this kind of offline version of simulation in terms of facial expressions. All the previous data has been looking at what happens online and why it would happen. We can really present the first evidence that we can start to see these effects occurring in terms of learning and in terms of prediction. These simulations are actually being retrieved and rerun in a predictive manner, not driven by actually viewing and simulating someone's expression, but being driven by having had that experience and having simulated them in the past, and then retrieving that when meeting them in the future. The other side of the PhD that I was doing, we wanted to pick a social cue to look at that was more subtle than, for example, facial expression or gaze direction. So although, for example, all these participants are actually unaware their facial muscles were being measured, their activity was being measured, they were actually pretty good at the end explicitly categorizing which faces showed which expressions. They're obviously very aware that they're seeing facial expressions. And similarly with the gaze cueing studies, although participants may not have been aware of the contingencies of the faces, the helpful faces and the unhelpful faces, they were obviously very much aware of the gaze direction element of the task. But just flicking through these two slides, we can pick a really very subtle social cue in pupil dilation. So the only difference between these two slides is that in this one, this lady has an average sized pupil, and in this slide, the pupils have become dilated. A pupil dilation is a really important cue for us as a sign of arousal, as a sign of engagement of attention and cognitive load. So in a social situation, it gives us an insight into somebody else's current internal state, but it's a cue that we are almost always not conscious of actually processing. So most studies in the past have looked at the effect of pupil dilation, have done something similar to this, where they've presented a pair of faces and they've said something like, which of these two faces is more attractive? And the answer, particularly if you're a male participant looking at female faces, is that you would pick the face on the right because it's got dilated pupils, but you wouldn't actually be aware of why you were picking it. And there's quite a good literature on pupil dilation effects. And consistently through that literature, the thing is that the participants are not aware of the fact that whatever their behavior is being driven, whether it's picking a more attractive face, whether it's choosing to interact with a certain person, whether it's perceiving an emotion differently. The participants aren't aware that it's actually this pupil size element that is driving that change in their behavior. We wanted to get away from, again, this idea of just looking at pupil size in terms of online processing, so in terms of what does it do to my behavior when I'm actually interacting with somebody. And we wanted to move on towards, could this very, very subtle social cue actually similarly be encoded, particularly because there's some evidence that suggests that to some extent it's a cue that's also simulated. So for example, this is just a study where participants are presented with different versions of different faces, and the faces either have dilated pupils, average sized pupils, or constricted pupils. And the participants who are, in the case of this set of data, men and women, are asked how attractive do you find these faces. So this is another UK lab. And in men looking at women, you get this linear effect. Basically, the bigger the pupils, the more attractive the faces. And the suggestion is that this sign of arousal, potentially sexual arousal, or just interest or engagement in a female face for a male participant is something that is interesting and positive to them. Interestingly, you get this quadratic effect in the female participants looking at male faces, where actually the most preferred pupils are those medium average sized pupils. And the suggestion in this study was that there was some sort of evolutionary reason for this. If a female and male meet and the male is showing all this pupil dilation very rapidly, the suggestion is that they're somehow interested in mating, potentially desperate to mate. Maybe they are not such a good mating partner for that reason. Another one, maybe they are aggressive, maybe
or stoned.
Or they're stoned. There's some kind of negative aspect that's underlying this sort of immediate sign of arousal. Interestingly, in this study, they then repeated the same experiment, just female participants and just male faces, but they split their participants using a scale that had people rate their partner preferences. So what your characteristics do you like in a boyfriend, basically. And they found a population, they divided their sample into 3 groups. So you had women who like nice guys, women who like what they call mover shakers, who are kind of your businessy sort of guys, and women who liked bad boys. And the women who like bad boys show this linear effect for male faces, and everyone else shows the quadratic. So there's something about particular people that they like this sign of arousal, they like it in particular faces. There's also some evidence to suggest that at different stages of a hormonal cycle, you may be more likely to approach somebody who's shown this sign of arousal. But the important thing being it's very subtle, it's implicitly or incidentally being processed. Also interestingly for us, this is a cue that does seem to be, to some extent at least, simulated by people who are seeing it. So this is a study where the researchers were showing that pupil dilation influences our perception of negative emotions. So if we see somebody with dilated pupils and they're looking sad, we actually perceive them as looking sadder. But the extent to which that occurs correlates with the degree to which the participants' own pupils dilate in response to seeing the dilation or perceiving the dilation in the image's pupils, so what we call pupillary contagion. This is also correlated with their levels of empathy. So the suggestion is that when I detect this very subtle cue of somebody else's change in effectively sympathetic nervous system activity. This triggers a similar embodiment in me, and that this is actually having a knock-on influence on my perception of that individual's current state. So this, of course, was interesting to us because we were interested in this idea of shared body states and how not only that might affect our perception at the time of an interaction, as in this study, but also how it might affect our long-term representations of another person. So in our studies, again, we went for this encoding and retrieval phase idea. So we have participants come in and they think that they're doing a facial vigilance task. So they're told, imagine you're airport security, you're looking at people walking past, you just got to detect when you see a new person. On every trial, the participant is presented with a blank screen and then a face. The face disappears and rapidly reappears again. And almost all trials are like this one, so the face reappears and it's the same person. But occasionally you'll get a trial like this one where the face reappears and it's changed to somebody else, and the participant's job is just to respond. We tell them it's about sustained vigilance. You're going to do this for 30 minutes. We just want you to be as accurate as you can be. The important thing is that when the faces appear in standard trials, so they're appearing and they're remaining there when after this blank period here, at this stage the face always appears with normal sized pupils, and at this stage the face always appears either with dilated pupils or constricted pupils. But again, importantly, just like our facial task, our emotional expression task, this is consistent within an individual. So when they're here, for example, will always appear with dilated pupils, whereas if I appeared, and obviously Wendy and I weren't in this task, we had different faces. If I was appearing, I might always have constricted pupils. So hopefully we get this learning effect where even though we hope our participants won't detect the pupil size changes, that actually because of the consistency, some learning is going to occur over multiple exposures. What we then do is, a few minutes after they've completed that first task, they're asked to make some judgments. How friendly is this person? And they're presented this time with just a static version of the face with normal sized pupils. So any difference in friendliness ratings, if it's different between those people who had previously had dilated versus constricted pupils, it has to be supported by some kind of recall effect, because here there's no pupillary cue. Everyone has average sized pupils. So for our first shot at this, we had, in all these studies again, female participants. And first of all, we had 10 female faces and 10 male faces. And what I should say about this data is that this all comes from participants who were naive to the pupil manipulation. A small percentage, so out of 30 participants, there are 25 participants in each experiment. And we might have, say, 30 participants overall, 5 of whom would actually have detected the pupil effect. Their data goes in the same direction. We just analyse the data from those people who are naive. So in this first study, we have 10 female, 10 male faces. Half of each sex group are going to be in the big pupil condition and half in the small pupil condition. And what's really interesting is we see this effect as predicted. Higher friendliness ratings to female faces whose pupils previously dilated than to previously constricted, and interestingly, the opposite for male faces. So previously constricted faces get higher friendliness ratings than those whose pupils previously dilated. So this is kind of in support of what's been found in the previous literature, but the most important and novel aspect of this is that this is a recall effect, so there is no pupillary information at the point when the participants are making these ratings. This is based on the fact that this information, despite its subtlety, has been encoded at that first exposure phase. But we had this question, why this in the male faces? Why does it go in the opposite direction? We get this significant interaction here. So for a follow-up study, we replaced the male and female faces with a set of male faces who'd been previously rated as being very trustworthy, and a set of male faces who'd previously been rated as very untrustworthy.
Who is that?
I don't know, it's some guy from a face set, but they all look like that. We did exactly the same experiment, so we have an exposure phase, and we have a recall phase, and we get them to provide ratings of friendliness. And the interesting thing is that we see the same pattern of data. These trustworthy guys are now treated in the same way that the females were. So if they previously shown up with dilated pupils, they're preferred by our female participants to those who previously appeared with constricted pupils, with the opposite effect occurring in the low trust faces. So the first interesting thing is obviously this static feature of the face, this sort of inherent trustworthiness in some faces, appears to influence the way that this really subtle cue changes our long-term representation of these faces. But also, presumably, particularly because some of the faces that fell into this group actually appeared in this group here, they were from the same sets. There's also some aspect of comparison, so there's some, the brain is going in comparison to this guy's really trustworthy, and I'm kind of glad that he's shown me, some sign of interest. So this was really intriguing for us. It showed that this sort of static and dynamic features are interacting, but again, at the center of it is this encoding of this really, really subtle cue. So just to really quickly show the last couple of slides of data, what we've always been interested in is this idea that underlying this is some sort of shared body state. So when I see somebody's, when I perceive someone's pupils dilating, even though I may not be conscious of it, it activates in me some similar sympathetic nervous system activation. And we have a version of this task in preparation using the eye tracker to look at pupillary contagion, which is quite a tricky thing to operationalize. But we just made the first pass of this by using galvanic skin responses and measures. So we had electrodes attached to our participants' fingertips, and we measured their GSR responses both during the encoding phases to these high and low trust male faces and the recall phases. The behavioural data for the ratings of friendliness looks like our data from when we didn't have GSR, so we replicate our behavioural effect. But the interesting thing is that if we look at, we can ignore this side, although we get a kind of, here we're looking at the low trust faces, and here we're looking at the high trust faces. So what we find really interesting is in the low trust faces we get a much larger galvanic skin response to those faces when their pupils become dilated than when they don't. So that's kind of what we would predict. What's interesting here is this actually flips, and one interpretation of this is that we're seeing some kind of violation of expectation maybe. So here we see this face that we don't like, and we see the sign of arousal in it. And here we see this face that in contrast we rather do like, and we actually see the opposite. We see a sign of, we sort of see the opposite of a sign of arousal. Someone's pupil is actually getting smaller once they've perceived us potentially. So this may explain this interaction between the two effects. But the important thing is within this particularly salient group, these low trust males showing pupil dilation, we're picking up this sign of sympathetic nervous system activity in the participant. And that's mirrored then at this phase when they're doing their ratings, when of course the faces are all appearing with normal average sized pupils. So we're seeing this reactivation of that effect in the low trust faces, with participants' sympathetic nervous system activity increasing in response to those faces who previously showed pupillary dilation. So we're just starting to touch on some kind of shared sympathetic system activity between a viewer and a target face, even though the viewer is actually not consciously perceiving the very subtle cue that's driving that activity. So the final thing we wanted to do was then to look at the direction this effect might be going in. So it's always interesting to know that an effect like this is occurring, but the question is, does pupil dilation make somebody more preferred, or is it that pupil constriction makes them less preferred? It's very difficult to tell without pre and post ratings. So exactly the same experiment with male and female faces, but this time we have the participants provide ratings of the friendliness of the faces before they do the exposure task, then they do the exposure task, and then they do the standard ratings task at the end. So we can actually work out the direction that these effects are going in. And what was interesting and very, I guess, very PhD about this was, of course, what we hadn't really thought about was you're going to get a mere exposure effect, probably, that to some extent masks the effect of the pupil size changes. But what was actually interesting about that is that all the faces become more friendly looking, basically, after this long series of exposures. We can see that the data are basically going in the same direction. So female faces who had pupil dilation, although the ones who have or show pupil constriction also become more friendly, these ones become more friendly. But actually, interestingly, the only group that go in the opposite direction are those male faces who show pupil dilation. So there's something particularly salient, similarly as we saw in the GSR group, about these less preferred faces showing this sign of arousal. And again, despite the fact that this is a very subtle cue, this appears to be driven by some sort of encoding. So what we generally put together in the package within the PhD was this idea that simulation is occurring not just online during interactions with individuals, potentially supporting some sort of online understanding of somebody else's internal state, but also that this simulation is being retrieved at a later time. So it's something that also appears to support perhaps the prediction of oncoming actions or the prediction of oncoming emotions. So I just have to say thank you very much to all the people whose data went into that talk, and thank you to you guys.
much. I really enjoyed that. I don't necessarily have a specific question for you, but some of the work that we've been doing downstairs has been on social contagion in people with autism, and also some a little bit like what you spoke about there in terms of trying to train people to generate these internal representations, these kind of efference copies, as like a predictive mechanism. So one thing that we've done is we used I'm not sure if you're familiar with the work by Anthony Hamilton and the eye gaze work. So we've done some work where we tried to use pro-social and anti-social cues to encourage imitation, voluntary imitation. So we regulated or modulated it quite successfully in a couple of occasions now. And so one thing that you showed in some of Steve Tipper's work was this eye gaze kind of like primed the direction, for example. So some of the work by Anthony Hamilton has suggested that, well, they control for attention. So it's just not just this directive, it's actually something about the eye gaze, or the pro-social, anti-social cue. So two comments really. One is, do you think what we've been thinking about is, do the eye movements themselves, not necessarily just the direction, the actual eye movement, so the saccadic response or the kind of the smooth pursuit response when they're making a direction, would that have a kind of a prime? That's kind of one comment. But secondly, I'd be interested to know what you're going to do now, because you've done a really good series of work there. So the second comment maybe first is an easy one. The first one, I don't know ourselves, we're going to try and do some...
Well, I had my visor on last Friday, so I'm well prepared. In terms of the eye gaze stuff, I'm not, I mean, the eye gaze wasn't really my area, and I started reading into it too much really when I was writing the introduction to my PhD, and then abandoned it. I've got a feeling that people have done work on what happens when you actually shift the position of the faces as well as changing the eye gaze. I think some work that Andrew Bayliss continued to do where they actually degraded the image so you get less information from the image. I can't remember whether Andrew's work, because Andrew's work was then also to do with this knock-on effect of how you encode that interaction. Whether it had more to do with the encoding, but I think the suggestion was that the positioning of the eye is the important driving factor. It's not just an intentional thing, it is something about actually simulating that physical eye movement. I have to say it's not something that I really read into an awful lot. In terms of where we go next, we're running some more experiments with my supervisor. We're interested in, to some extent, boosting mimicry, so looking at actually, as you're interested in, what states can we put a participant into that might actually affect the degree to which they show mimicry effects. But also, in terms of understanding what the mimicry effect is it just something to do, can we tease it apart from the effect of affect, basically? So we know that facial expressions are rewarding and punishing. Is it just that facial expressions provide us with, for example, a rewarding smile that then activates some kind of affect program that includes activation of the zygomaticus? And there was some experiments in my PhD where we looked at doing similar paradigms but using non-facial affective stimuli, so pleasant and unpleasant animals, where we started to show that these kind of effects do appear to be unique to facial expressions. There's some work from Paul Liebenthal that showed similar things. So it'd be interesting to go down that route and actually try and come up with paradigms where we can start to pull apart what is actually embodiment and what is just an affective response to seeing a particular stimulus. In terms of the pupil size route, again, we're kind of interested in whether we can boost this effect by looking at what states are likely to make people super sensitive to these kind of subtle social cues. Also, it would be nice to move away from just using the same kinds of rating responses at the end of the task. But of course, that element of pupillary contagion is interesting, because although responses to Francis's question, we don't really see within our fairly narrow sample much in the way of influence of individual differences. The suggestion would be, given we see, for example, mimickers and non-mimickers, that really there is a great deal of individual differences between people in terms of how much you're likely to simulate somebody else. And it would be interesting using that pupillary contagion technique to look at what happens to our recall effect between those people who show pupillary contagion and those people who don't.
It's a nice diagnostic probe into the social brain, isn't it? You start looking at the boundary conditions that you are for autism. It's a good experimental example of one end of the spectrum. And the subtlety of these measures is quite impressive. I So how long is it learning? This learning takes how long?
About 30 minutes.
So it's quick.
Yeah, I mean, you're getting multiple exposures obviously to every face, so each face 10 times in the pupils. One thing that's actually interesting in terms of those pupils as well is, as you were saying with the gaze, what are they actually encoding?
Yeah.
And one of the questions I'd really like to answer is, can you actually get the same effects if you provide a reason for pupil recontagion? The brain might detect like a change in light levels that would explain why someone's pupils will change in size, whereas in this case, of course, the assumption the brain is presumably making is that change in pupil size is to do with them, is to do with you. So it'd be interesting to manipulate that and see whether the brain actually detects the difference between a physical change and a psychological one.
Sure. I'll ask another question now. Sorry, I've gone.
A couple of points really. One was, almost picking up on what you were saying just then about individual differences and having mimickers and non-mimickers. And I was thinking at the time when you were talking about that that's quite an interesting thing. I'm wondering whether, because people vary in themselves as to kind of talk about people who have very expressive faces and don't have expressive faces, and I wonder how this kind of link all kind of comes together, and whether people who themselves have more expressive faces, are they people who will mimic more or less? So like some
Absolutely.
looked at, there's questions around that, and that's kind of whether there's something about your participants as well as about the stimuli that you obviously started to kind of look at.
Look into, and also that element of how aware people are of their own physical states. So it's interesting to look at those people who have sort of good proprioceptive awareness. That's something else that would be interesting to follow up on as well.
You mentioned throughout your talk that this response seemed to be potentially underpinned by like an automatic response or lower level processing. Given that Steve Tipper and Patrick then published that paper which showed that these lower level processes are under top-down control from the primes they use, the colour primes. So we've got a couple of papers under review at the moment that shows, we hope if they get published, we think we've shown that we can modulate how these lower level processes that code biological motion, particularly velocity characteristics, and we modulate it through attention, so directing attention using something like Patrick, but also we did a belief manipulation where you can actually prime that system via a verbal belief mechanism prior. So you did your prime, so we prime it via a belief manipulation, a verbal prime. So it's independent of kind of visual stimulus, and it's purely this top-down control, I guess, not necessarily attention, but top-down control. I wonder whether you could comment whether you, from your work in Steve Tipper's lab, and whether you think that mechanism is a trainable, top-down, bi-directional process, a little bit like the CCD Hayes model, ASL model, or you think this intelligence that is automatic?
I mean, it certainly seems to differ as well in terms of things like object affordances versus action affordances towards other human beings. I don't know, I mean, standing up here, that's the honest answer, I'm not sure I can comment. But certainly looking at things like facial mimicry effects, these do seem to be very automatic processes. So they are things that even if you purposely ask your participants to inhibit the response, you will still measure facial mimicry like activations, just weaker activations. So I think to some extent this is an automatic process that can be inhibited, and certainly some work that, I don't know if you know the work that Steve did with Peter Cannon on facial mimicry, where similarly they used in the same way that we did, situations where attention is actually allocated to the expression itself versus allocated to, in this case, a non-social element, so the coloured mask level of the face. You still see facial mimicry effects regardless of whether the emotion is task relevant. But actually what's interesting is that you see a sort of inhibition of that facial muscle activity. So generally the muscle activity is inhibited, but the pattern of activity is exactly the same. And we've, in other studies, got some data that's quite similar to that. So I think that these are very automatic processes, but it's just in the same way as like my brain is capable of inhibiting my embodiment of the ability to grasp this jug if I want to grasp that cup. I think this is something that is presumably more difficult to do with these very socially salient stimuli, but I would imagine that kind of more conscious controls can prevent these sorts of mimicry responses, but to some extent they're always there. They are, because of their automaticity.
Obviously, you were saying what was novel about your work is the fact that you've been looking at it offline. But it's still quite a short period after. So I'm wondering how long does that last?
Well, this was
Changing forever.
This was another study that we would like to have had time to do, where either we did, I mean, logistically doing a test retest is always kind of difficult because we get participants back in. We would like to have done something where we put a filler task in the middle, so we have to do something else, maybe face-based, with a new set of faces, and then we get them to do either a recall task or one of these re-exposure tasks to the facial memory. I would have thought that the effects would last. I mean, there's some evidence to suggest that encoding should actually be stronger with a period between encoding and retrieval, so we actually might get stronger effects, but it's very difficult to say. And I think it would be really nice.
6 months, you really did
That's right.
judgments over a long period of time.
I think you've got to have, I think to some extent you might need to boost not just the simulation itself, but also the reasons for encoding the faces.
Yeah, I was just going to say, because presumably it kind of gets, you have so many social interactions with different people, and presumably that, as you say, the frequency of encountering, but it's going to essentially change.
I just want to sort of follow up really, as well as how long these things last for, whether or not there's anything out there that sort of shows how long would it take for a trustworthy person to become untrustworthy as well? Because it seems like the data you're presenting is, the same way, a lot of this research is done. One person is always in a particular state and another person is always in a particular state. Presumably after your 10 exposures to yourself being helpful, you're seeing this effect. But if you gave the 10 exposures, presumably it's now been learned, but then on the last 2 you were to be, so do you get kind of a recency effect, or are you just a slightly flawed human at that stage, but you've generally been helpful throughout the times?
Well, I think I guess one thing would be that it would depend how you actually measured the trustworthiness. So this one suggestion would be that actually you would see a greater mistrust effect, if you like, if you prime somebody as being somebody really helpful, and then suddenly you flip them over, because of course this violation of expectation. It's going to cause a marker which is very powerful for that particular effect. So you might get your very, very rapid, how quickly you would see a change in somebody's level of trustworthiness if they start out with a neutral stimulus. I don't know. I mean, of course, with this, we actually tried to do a version of the facial mimicry task where we actually embedded the neutral faces in amongst the emotional ones. So participants were doing like happy, angry, or neutral, or they were doing male and female, but every so often they see a neutral version of the faces. For exactly that reason, we wanted to sort of see when does this simulation start paired up with this face, so it's activated for the neutral version. Interestingly, we actually got what hinted at a contrast effect. So faces that had smiled when they were seen as a neutral face, you would actually see less zygomaticus activity than for a face who previously frowned, presumably because those faces, in contrast to their emotional versions, appear to be more angry or more happy. So you actually see this kind of interesting rebound effect, and it kind of illustrates that it's quite difficult to do those sorts of tasks, where with measures like these you start to look at kind of these sorts of changes, how rapidly they evolve. So I don't know the answer to that, but some work that's currently going on in Steve's lab, looking at data cube behaviours, does suggest that this kind of violation of our expectations of an individual is quite important. So as you were saying, if someone who we think of as being trustworthy suddenly becomes untrustworthy. And I know there is some scanning data out there, so they started to look at this kind of updating and where that's occurring in the brain. I think that was quite rapid as well, but how that.
Okay.